Hello and good evening to anyone who is watching anywhere in the world. Today we have the very first international forum on autoimmunity in COVID. Now this may be a topic that we are not very familiar with, but the important thing to say is that viral-mediated autoimmune responses are very, very common with multiple diseases. What we've got here today are a group of experts from different parts of the world who are focused on this area in the context of COVID-19. The big question to ask is, why have we not got the answer to this as yet? This is such an important area and it requires all the expertise that we need. And so I have the pleasure of introducing to us our three extra experts from the world. We've got Dr. Neil Bodie, we've got Professor Bruce Uhal, and Dr. Jean Amiral from France. So guys, thank you very much for coming. Oh, sorry, I think I had unmuted you. Now I've got you here. So, yep, can everybody hear us? Just say some words.
Yes.
Good morning. Excellent, excellent. Okay, what we'll do, so I'll just ask you guys to introduce yourselves, and I'm going to start with Bruce. Do you want to go ahead first and let us know who you are?
Sure. Hello, everyone. I'm Bruce Uhal. I'm a professor of physiology at Michigan State University. And I've been working for most of my career on lung injury mechanisms, and in particular, the role of the angiotensin system in the lungs, local angiotensin system in the lungs, in lung injury. And for about 16 years, I've been working on the enzyme ACE2, which we now know is the primary receptor for the SARS-2 coronavirus. But my work has been focused more on the role of ACE2 in the normal lung physiology, where it's been shown to be protective. So that's my perspective on this. And more recently, as we learned that ACE2 is the receptor for the coronavirus, I've sort of unwittingly become an expert on SARS-2 infection through this receptor. So that's my story.
Wonderful. Thank you, Bruce. And Neil, do you want to go next?
Yes. Good morning from Los Angeles. I'm the CEO of Paradigm Immunotherapeutics. We're working on, we started originally on autoimmunity and then transitioned to infectious diseases, and right now concentrating solely on COVID-19. So again, I've got a lot of experience, decades in studying autoimmunity, and then we started looking at how something called antibody-dependent enhancement made infections worse, and then I have been studying SARS-2.
Wonderful. Thank you very much, Neil. And what about Jean?
Yes. Good day, everyone. So I am biochemist, and I have a doctor in medical science, and I have been working especially in research in hemostasis, thrombosis, and fibrinolysis, and I dedicate a part of my work to autoimmunity-associated thrombosis, especially Repeal-induced thrombocytopenia and some autoimmune disease which are associated with some infections, such as antiproteinase antibodies, which develop in some patients with varicella chickenpox. And so focusing on the new disease, on COVID, and trying to understand the mechanisms, there were very surprising features. And some of these features present some of the autoimmune complications seen in the field where I was working. This is why I focus on this, and my idea is that autoantibodies to ACE2 can develop in these patients, and now I am trying to develop an assay for testing these autoantibodies in case they are present.
Wonderful. Thank you very much, Jean. And just to give a general concept is that for people who may not quite be aware, when we speak about autoimmunity, people would think about diseases like rheumatoid arthritis or systemic lupus. And all it is that the antibodies that would normally fight against viruses or bacteria or any foreign pathogen that enters the body these antibodies, instead of just targeting something else, end up targeting a part of the body. And it could be the kidneys, it could be the heart, it could be the joints. And this is what an autoimmune disease is about. And so the overall principle is, could we be having autoimmunity in the context of COVID-19? And so as a general question, how in the world has this been not answered by now? Anybody want to go first?
Yeah, I'll mention that because, I'm sorry, Bruce, something that another clinician brought up is when you're, fighting the virus and it's a pandemic and your ICU is overloaded, it's very hard to be conducting research studies. But there are a lot of people that are long haulers that, have clinical problems weeks, months afterwards, and those people need to be studied in areas where now the pandemic is down, like in the New York City area.
Yeah, and we're going to come to that in quite a bit of detail. Any thoughts on this, Jean, in terms of why is it that we've missed this question from a research point of view?
So on the research point of view, what looks striking is how the disease evolves, at least in some patients with long-lasting symptoms. And this evokes the presence of autoantibodies, especially because many organs can be targeted, and AC2 is rather ubiquitous. So it means that if autoantibodies are present, the disease can strike in different parts of the body. And it is what is observed in some patients. So this is the basis of the development. Then the most probable candidate is ACE2. And I have been working in order to find a source of ACE2. And now I have ACE2 through a partnership with an Austrian company, which allows to work for developing an ELISA. And the next step is then to try to validate this ELISA with well-selected patients.
Wow. Now, let's just clarify something, because some of those concepts you mentioned there are quite complex things. So before I'll even go into those details, and I think both Jean and Neil are the experts at the development of technology to assess this. Bruce, can you give us some kind of idea about what really is this ACE2, and how does it fit in this whole puzzle with regards to COVID-19?
Sure, I'd be happy to. Can I share the screen?
Certainly, yeah.
Show some figures. If I'm looking at my Chrome tab, okay, here we go. Can you see a figure now entitled "Roles of ACE2 in Lung Fibrosis"?
Yes, I think we can see that.
Okay, this is a summary slide that I show whenever I give a research lecture that summarizes some of the information that we discovered and published over the last 15 years. As I said, my research over the years has been focused on lung injury and fibrosis and the role of the angiotensin system in lung injury fibrosis. And that's summarized here. This line on the top summarizes about 15 years worth of work in various publications. And what we showed was that during lung injury, the gene for angiotensinogen gets turned on in the epithelial cells, primarily in the lung, to make angiotensin 2 locally in the lung. This is extravascular angiotensin 2, a local tissue angiotensin system, which is pro-apoptotic for epithelial cells of the lung, which of course is a very bad thing, and also pro-fibrotic. And more recently, in about the last 15 years, we've been studying the role of ACE2 in this whole system, which is protective, because the normal function, one of the normal enzymatic functions of ACE2 is to remove this single amino acid, the phenylalanine from the C-terminal end of angiotensin II, thereby reducing angiotensin II and reducing apoptosis and fibrogenic stimuli in the lungs. Angiotensin is also pro-inflammatory. So ACE2, simply by removing angiotensin II, degrading it, protects the lungs against these effects. But in addition, it produces the peptide angiotensin 1 to 7, which itself is protective through a receptor called MAS. And one of the interesting things we showed previously was that in the lungs of patients with idiopathic pulmonary fibrosis, this is data obtained from biopsy specimens from human lung tissue. Here is ACE2 mRNA and enzymatic activity in normal human lung. But in the lungs of patients with fibrosis, ACE2 mRNA and protein and enzymatic activity are reduced. So over the years, we were trying to understand what is it that reduces ACE2, this protective enzyme. And in fact, our hypothesis was that this reduction of ACE2 might be one of the mechanisms that's leading to the fibrotic response in these patients. So that's the perspective that I had coming into this. And of course, more recently, now we know that ACE2 serves also as the receptor for the SARS-2 coronavirus, as it did for SARS-1 about 12 years ago. And so if you look at this figure from this nice paper from the group in Göttingen, Germany, ACE2 binds the coronavirus spike protein, the receptor binding domain. And then it is endocytosed, and we know that there is a requirement for infection, not only for ACE2, but also a protease or proteases. TMPRSS2 protease has been required for priming of the S1 subunit of the spike protein. And infection can occur by that route through TMPRSS2, or some people also believe cathepsins, that once an endosome is formed, cathepsin activity is required for proteolytic processing of the spike protein. There are some data to suggest that ACE2 itself is also proteolytically cleaved by some enzymes. So this now is the process that ACE2 contributes to, that I'm now focusing on with our colleagues. So
it raises a really interesting question, and just takes us straight into the issue about the long haulers. One of the biggies is that there is so much lung disease post-COVID, even for people who didn't end up on intensive care. They seem as though there are still fibrotic changes that are occurring. Could that just be from the damage that is happening to the ACE2 receptors on cells?
It could be. We know from a variety of animal studies that if ACE2 is either reduced or inhibited simply by applying, for example, a competitive inhibitor of ACE2 and squirting it down the tracheas of mice, they will develop lung injury and a fibrotic response just by specifically inhibiting the enzyme ACE2. So if the binding of the coronavirus is compromising the normal protective function of ACE2 as an enzyme, that in itself could lead to lung injury. But that's still at the level of theory.
Yeah.
And there aren't any convincing demonstrations that mechanism alone accounts for the damage that we see in the lungs of COVID-19 patients. But it certainly theoretically would be expected to be a contributing factor.
Yeah. Any thoughts on that, Jean, in terms of any experience with that kind of situation?
No, I am more specialized in hemostasis, but what we can add to what Bruce said is that AC2 is, of course, very important for lungs, but not only for lungs, because it is present in many organs, and especially liver, kidney, probably brain, and also in endothelium. And all the different organs or tissues which have been affected by this disease are concerned by ACE2. So this is a very important point. The infection, the viral infection, has another impact. It is the shedding of ACE2. Not only the blockade, but also the shedding. It means that during infections, the density of ACE2 on cell surface is reduced. So this is very interesting, especially because it is present in many organs, in many parts of the world, and also in intestines. In intestines is one of the organs with the highest density after lungs.
Any thoughts on it, Neil?
Yes, I mean, looking from an immunological standpoint, one of the things that surprised me is that the binding of the receptor binding domain to ACE2 is incredibly stable. I mean, you know, it sticks once it binds. You know, I've seen affinity models using Biacore or BLI, and after 30, 40 minutes, it's still a stable complex. And so that means there's going to be plenty of time for the immune system to see that. And both ACE2 and the spike protein go through conformational changes. In other words, the three-dimensional shape actually changes, and that's something that can activate the immune system. And that's something I don't think has been studied very much at all.
Yeah. So I think one of the things that I will share from our perspective and our research that we've been doing, Bruce, is that we proposed that the critical piece of the puzzle in the context of autoimmunity, we propose is related to serum ACE2. And just to clarify that concept is that for some reason in hypertension, heart disease, renal disease, the body cuts off the ACE2 from the surface of the cell and it floats in serum. And as Bruce has said before, we don't even know how much activity the enzyme has in serum. So we're not quite sure why the body does this. But here is the interesting question, and this is what we have proposed as the autoimmune question is, if you have serum ACE2 floating in the blood and the person has a viremia, so the virus is in the bloodstream with the spike proteins, if you have the serum ACE2 binding to the spike proteins before this virus is picked up by the immune system, could you then have a situation where the immune cell perceives that the ACE2 is a part of the spike protein and produces antibodies to it? And that's the essence of the autoimmune theory that we've said, and it's why we think you have this predilection for older people and people who are overweight because they have higher levels of serum ACE2, and it just seems as though it makes sense. But that's our hypothesis, and I'll allow you guys to share your perspective on how you think the autoimmunity could work. I'll start with Jean. Any thoughts on your perspective as to how autoimmunity would occur?
So the first point is what Neil said. It is a very strong interaction between the spike protein, at least the RBD domain, and ACE2. And it is very strong, and it has been demonstrated with recombinant ACE2 and recombinant spike protein or RBD. So the strong complex, which can last in the body for a long time, is an ideal situation for inducing an immune response. And the immune response can be deviated to the self component in addition to the viral component. And this is something that it is noticed in many mechanisms which can induce autoimmune disease. Now, probably that not all patients will develop antibodies, even if they have the same autoimmune stimulus. maybe some genetic factors can be involved for favoring the development of immune disease, but we have no data at this stage. This is only a hypothesis. And there was recently a paper from an Italian group who also suspected the presence of genetic factors for inducing autoimmune complications in patients with COVID.
Yeah.
Now, the two things can contribute. One can be the genetic predisposition. The second one can be the intensity of the immune stimulation. Both can be combined. Eventually, a third factor could be the presentation, the complex presentation between the spike protein and ACE2. Some specific presentation can favor the immune response, as we have also examples in other domains. Especially, I had experience in heparin-induced thrombocytopenia, where a specific conformation presentation of the self-component, which is PF4, and the foreign component, which is heparin, has to be formed.
Mm-hmm. And Bruce, in terms of the question about how well ACE2, even serum ACE2, binds to the virus, we don't actually know, do we?
Well, there are some data primarily from the SARS-1 coronavirus that have characterized the binding of ACE2 by the soluble ACE2 to the virus, but there are less data to characterize the binding to the SARS-2 coronavirus of the soluble ACE2 that gets released from cells. And remember, it's not the entire protein; it's the extracellular domain that has been cleaved off. So, in a strict sense, it's not the same protein that the virus binds to when it's attached to cells. It's
That's a very good point.
A cleaved form that does not contain the entire sequence. As Dr. Bodie alluded to, we know from in vitro studies of binding that much of which is based on recombinant proteins, I should point out, that conformational changes occur to the ACE2 when it's bound up to virus. So if you can envision the virus binding to the soluble ACE2 in the serum, it's quite likely that there are conformational changes that may generate new epitopes that would be the target of autoimmune antibodies if our theory is correct. And so another point that may be important is we know almost nothing about the fate, half-life and fate of this soluble ACE2 virus complex that's floating around in the blood. What happens to it? Is it ingested by, say, macrophages? A testable hypothesis, but let's say it is ingested by macrophages, and those macrophages start degrading this complex of ACE2 in the virus. Then there may be generated degradation products that also present as new epitopes for antibody generation. So many things that we don't know yet about the ability of the soluble ACE2 to bind, how long does it last with its bound up to virus, and what sort of molecules are generated as it's being dealt with by whatever cells deal with this complex throughout the body. I mean, you could envision this complex depositing in various organs and being dealt with differently depending on the organs that sees this complex deposited. So lots of things we don't know yet about how this system is working.
Yeah. And Neil, any thoughts with regards to that?
Well, this is a little bit off subject, but it still has a lot of impact on what we're talking about. And that's the antibody-dependent enhancement. Now, in classic ADE, you get virus infecting macrophage type cells, and they reproduce in those macrophage type cells and enhance the infection. What we've found testing against live SARS virus, and it's now been published, is that SARS-2, unlike SARS-1, does not seem to be able to have a productive infection in macrophages. So you don't see classic ADE. But what we found, and what others are finding out, is that binding to complement. So around day 12, when a lot of patients become critical, immune complexes form, and those immune complexes bind to the first part of complement C1q and cause complement activation, and that can enhance the infection. So there's two mechanisms of ADE. The first, the classic, may not apply to SARS-2, but the second, complement ADE, that really probably contributes. And I think it also contributes to the thrombosis that we're seeing, the blood clots and things that we're seeing in COVID-19 patients. And one thing that's very interesting to point out before I give up is the largest pool of the activating Fc receptor, FC2A, that causes ADE is found on platelets. There's about 20,000 per platelet. There's 500,000 platelets per microliter. So in people and non-human primates, that's the main source of the activating Fc receptor that can trigger ADE on platelets.
Wow. So there's so many unanswered questions. And so what we're going to focus on now is what I call the low-hanging fruit. The low-hanging fruit is trying to find an answer in the context of COVID, not just about the seriously affected patients, but the long haulers, the people who have had the infection and are left with persistent smell abnormalities, taste abnormalities, fatigue. There's a whole range of symptoms. And within the context of autoimmunity, how does that fit? And I'll probably start with you, Jean, because you're probably the closest to finding some answers to that. Any information or anything you want to share on that? Jean, can you hear us?
Yes, the comment I could make, it is the comment to Bruce, is that the soluble ACE2 has probably a very strong binding to the virus, to the spike protein, and it's very strong. And maybe you heard about this research from a French research center who is using an ACE2 peptide for decreasing the infectious capacity of this SARS-CoV-2. And only a small part of the ACE2 peptide can be enough for preventing the virus to infect the target cells.
And so in terms of long haulers, as we shift now to what we describe as long haulers, these are the patients who have long-term symptoms. What is your research focused on at the moment, Sean?
So for the long-term symptoms?
Yes.
Yes. So what I am looking now, it is to develop an ACE2 ELISA for testing autoantibodies. And probably it should be useful to not only test autoantibodies, but to test in patients the soluble forms of the different factors of the renin-angiotensin system and see if there is any impact during the infection. But the major goal is to have this ELISA for autoantibodies to ACE2, and then to have patients who have lasting symptoms for more than 3-4 weeks, and to check for the possible presence of autoantibodies to ACE2 alone or ACE2 combined with RBD or with spike protein.
Wow. And so in terms of the similar thing, are you looking at a similar thing, Neil, with regards to?
Yes, I mean, we're working on a chimeric ACE2 antibody, and we're modifying it so that it's not going to trigger what I discussed, complement ADE. And then we're looking at that as a prophylactic. And as part of that research, we want to see if we're generating any type of immune response to the ACE2 on that therapeutic. But that's going to, we'll also be looking at ACE2 itself and making sure that, we're not doing any harm in that area. But I think that that's something that is a very valid point on these long haulers, and something we have to look at. And I think the fact that steroids, dexamethasone, methylprednisolone, there's been a number of different steroids. When they're used after the viremic stage, they increase the survival by 30 or 40%. That says a lot that there has to be obviously an autoimmune or immune-mediated component to observe that type of response.
Absolutely, absolutely. And Jean, in terms of the screening that you're looking at for the long haulers, how far are you away from being able to
So at this time, I just am developing the ELISA. And then I plan to have collaborations with some clinical centers. I have already different contacts in this field where they are following patients with these long-lasting systems. And the first step is to focus only on these patients and eventually to extend to other patients after.
Wow, wow. And Bruce, we were recently with Neil having a discussion with Josh. What are your thoughts with what that was about?
Well, the first thing I think that we need to do, and we were talking about with Josh, is to attempt to get some feel for the titers through say dot blot assays, that if there are antibodies present. We have no clue yet how concentrated, what the titer might be in the serum. So we're thinking as a preliminary approach to begin answering if there are autoantibodies against ACE2 and or the complex of ACE2 and virus, to do a screen such as Sean has alluded to with recombinant ACE2, and first look at a variety of dilutions of serum to try and get an idea. If there are antibodies there, the first thing we need to know is what sort of titer, what sort of concentrations, and how far do we need to dilute the serum down to see such a thing. And then it may arguably be useful eventually to go to using fragments of ACE2 and or the spike protein on peptide array studies to try to look at all regions of the proteins, so as not to exclude any possibilities. For the possibility we were talking about before, where there may be degradation products of either ACE2 and or the spike protein or the complex that are produced if they are degraded by various phagocytes, for example, that could be generating epitopes for autoantibodies that might not be observed if one uses, for example, a recombinant ACE2 protein in a plate or a dot blot type assay.
Yeah.
So that's what we're thinking as general approach to begin looking for this theory of immunity.
So I think just to clarify, because some of the concepts that are being discussed here are very complex research ideas, and just to break it down a little bit, and you guys can tell me if I'm about right in terms of the thinking, is that we are thinking that after the infection has passed, for many people, they still have autoantibodies that have been created. We're still not sure what kind of autoantibodies, but our suspicion is that they have autoantibodies created which are causing long-term symptoms, okay? It's one thing to be able to identify that this is the case, but even if we identified it, would we be able to do anything about it if this is what has happened? Any thoughts on that, Neil?
Certain medications, azathioprine tends to concentrate more on B cells than on some other areas. But azathioprine is a chemotherapeutic agent. I mean, I only use it in cases where I've proven an immune-mediated disease because of possible side effects. So, the ideal way would be to try to use maybe the B-cell type of drugs that they have for lupus, where, they're trying to decrease the activity of B cells. In extreme cases where people are having life-threatening infections, you could have something where it causes the lysis of B cells. So B cells are what produce antibodies. But we have to identify first, everything that a doctor recommends, to spill up as a combination of risk versus benefit. So before we recommend a drug that may have serious side effects, I mean, we're cautious already about the use of steroids in infectious disease. That sort of goes counter against what we normally think of. So, the use of these drugs would be after the viremia is cleared so we don't make these patients worse, and then closely monitoring them for the side effects and try to selectively concentrate on the B cells and the antibody formation.
Yeah, and so it raises a point that I think Jean had said in the past when we'd spoken before. You have found, Jean, that in quite a lot of patients in your previous work on autoimmune diseases in the blood, that the autoantibodies tend to automatically disappear. Is that about right?
Yes, usually autoantibodies, so they are very active at the beginning when they are developed, and the disease can last for 2-3 months. And autoantibodies then progressively disappear in most of the case, and after 6 months, they are no more found in blood of the patients. This is true for drug-induced antibodies, but also true also for some autoantibodies induced by viral infections.
Yeah, so this is it's an important issue, because what we would hope is that people will lose the symptoms will disappear as time goes past. And I'll mention, because we've just been working on this, Bruce, is that we're developing a concept where a simple drug could make a difference in COVID-19 if this is part of the autoimmune, and that's aspirin. And we've just written a very short letter to the editor where we're pointing out that this may be of value even for long haulers if this is potentially an autoimmune response. So we do have to start thinking, what would we do if this was proved? And Bruce, just from a scientific point of view, because you understand the research area much more, how difficult is it going to be for scientists to accept that autoimmunity could be the cause of COVID-19?
Well, as someone who has spent much of his career writing grants to various agencies like the NIH and American Heart and so on, it's the job of reviewers and other scientists to be skeptical right from the start. And in fact, we have already received some not only enthusiastic reactions to our autoimmune theory, but some skeptics who have said privately, oh, I just don't believe this, blah, blah, blah. So I would expect there to be some healthy skepticism, even if we do obtain evidence for autoimmune antibodies. There will be skeptics that will say, show me some proof in some sort of a model that these autoimmune complexes are indeed responsible for the organ damage that you're proposing they are responsible for. And that kind of scientific proof may require the use of some animal modeling, perhaps some human organoid models to convince people that, yes, this type of autoimmune antibody that we've demonstrated, say in IgM or something, that in this model, it does result in damage that could explain what's happening in these patients. So I would expect this sort of skepticism. This is just the way science works. There's going to be people that say, well, prove it to me. And it may require some animal modeling, for example.
Yeah. And Neil, any thoughts on how difficult it's going to be to convince the scientific community?
Well, again, I mean, we've even changed the name of autoimmunity in the last 20 years to immune-mediated because of those skeptics that Bruce just alluded to. So, they've argued that the term autoimmunity may not be as appropriate as immune-mediated. So there's an example of how, skeptics have changed their outlook on this whole area in the last 20 years. And I'll just point out that there's sort of a double-edged sword. As these autoantibodies decrease, so are antibodies to SARS-2. And there's been several studies that show that the antibodies to SARS-2 decrease substantially over months period. And there's now, though the rare, been instances of reinfection. So it's sort of a double-edged sword. And certainly because you get an amnestic or memory response, if you get reinfected, does that mean that you're going to be more likely to have autoimmune problems after the second infection? These are things we just don't know, but are things we need to answer as quickly as possible.
Yeah. And Jean, from your point of view, is even if you demonstrate autoimmune antibodies, it could quite reasonably be argued that it's not necessarily a part or relevant to the disease.
It is difficult to say. We need first to have tools for exploring patients and to see if the patients who have the lasting symptoms are those with the antibodies. This is a very important point. And this is my objective, to have this assay available with the global protein, and then to try to have this kind of testing in order to find a relevant group. My idea is that if autoantibodies are present, they are more harmful on ACE2, which is on cells, which is the receptor, which is exposed by cells, than for the soluble form, because the soluble forms probably will be eliminated rather rapidly through macrophage or other elimination systems or other clearing systems. But if antibodies target ACE2 exposed on cell surface, then the immune response is deviated to these cells. And then this infection can have three harmful effects. One is the infection itself by destroying the cells. The second one is by impairing the renin-angiotensin system, inducing inflammation, fibrosis, apoptosis, as Bruce presented it. And the third one eventually is to deviate the immune response to cells themselves and not only to the viral infection.
Wow. Listen, I think as we're coming to the end of this presentation here now, what I want to ask us to look at is, if we had almost a wish, if you had to ask for anything at this point in terms of finding solutions for COVID-19 in the relation to your work, what would you ask for? And I'll probably start off with Neil. I'll start off with Neil. What would you want at this point? You have all the wishes, all the power in the world. What would you want?
Adequate funding. I mean, that's really what this comes down to be able to fund you and Bruce and other people and Jean that are working in this area. But to get answers, we have to do research. To do the research, you have to free up the funding. Most of the funding from the federal government right now is concentrated towards vaccine development and monoclonal antibody development. But we need to free up some funding to be able to study these effects from the long haulers.
Absolutely. And Bruce, what would you want?
Well, to begin with, I would want $3.5 million to research, which is about the budget of the NIH grant that I submitted a month ago. So we shall see. But no, I agree with Neil. I mean, science costs, but from the outset, if we are able to obtain some preliminary data that even some rudimentary data says, oh, look, here's some evidence for autoantibodies from this limited number of samples that we've been able to gain access to, and we have a simple screen that looks positive, then we have some preliminary data that we may be able to use to raise the enthusiasm from those who have monies available to devote to further experimentation to try to prove and or disprove this hypothesis. Actually, we need, I think we're right that there's autoimmunity involved, but we may be wrong, and we need to prove it either way, just for the benefit of all in the world. So certainly funding, but you move forward with what you can, what you have at your disposal until the funding comes through. So however we can go about it with resources that we have at the moment, we're working to move forward to test this hypothesis.
Yeah. And Jean, from your point of view, what would you want? What do you need?
So, on my side, I am not working in fundamental research, but in practical research. And my goal is to develop and validate the assay, then to propose the assay. And for this task, what I wish is to have some partnerships. I can have some partnerships on the clinical side, some partnerships for developing the assay itself. I have already some of them for having access to the raw materials which are needed, and then eventually to have also research partnerships for completing, if we find something which is of value, to complete the investigation of the epitopes. But because maybe we can have one response, we can find something yes or no type response, but the work is not terminated at this stage. We have to understand if it is the optimal target antigen and which kind of epitopes, then it is a more complicated job. And in this case, I would like to work with more fundamental groups for achieving this objective.
Wonderful. Yes, and I think that probably brings us about to the close. And gentlemen, I think what has clearly come across is that in order to do research, proper research, it costs. When we think about how much of a financial impact this COVID-19 has had on the world, what we are talking about to find the solutions are a fraction of the cost that it would be. Because if autoimmunity is demonstrated, without a shadow of a doubt, we have the capability in medical science to protect those who are at risk, to treat those who are suffering with the disease, and to manage people who have long-term symptoms of the disease. I think that there is a chance for us to find solutions. And I would like to thank again the whole panel here who shared some very, very interesting insights into autoimmunity. I hope this was of value to everyone. Thank you very much to you guys, and I wish you all the success going forward.
Thank you.
Thank you.
Wait, John wants to say something. Go ahead, John.
Yeah. Yes, just I would like to say that if autoantibodies are confirmed in this COVID-19 disease, this has an impact not only for diagnosis and for managing patients, but also on the vaccine development strategy. Because,
very true.
Yes, if the vaccine is based on spike protein, for example, and if spike protein can form strong complexes with ACE2, this can be a cause for inducing side effects such as autoantibodies. So, of course, this is only a hypothesis and this needs full demonstration, but this risk is present and this can have some impact on the strategy for developing the vaccine.
Wonderful. Very, very important point. And you're absolutely right. If we don't prove autoimmunity and it is part of the disease, it will absolutely create havoc for people who are vaccinated. So yes, we need the answers, we need the help. And again, thank you very much. And I'm sure we'll have further and conversations in the future about this very, very interesting topic. Thank you very much, gentlemen.
Thank
Thank
you.
you.
Yeah, thank you.